This video will be on gene annotation. This is the next laboratory project we'll begin. And first I'm going to start off by talking a little bit about how genomes get sequenced, and then the new field that has developed called comparative genomics. I'll talk a little bit about the research question that we will go into in lab and talk about the evidence that we will use for gene annotation. So most genomes these days are sequenced via what's called shotgun sequencing. And we've talked about the actual process of sequencing. All of the sequencing technologies take that entire genome and cut that genome up into little pieces. All of those individual pieces are then sequenced, and at that point we try to compile the genome back together again. So once those sequencing reads are completed, then we can find the regions where the sequence overlaps and start to assemble those into what we call contigs. Just as an example, here is the Gettysburg Address broken up into lots of little pieces, and you can look for the overlap and begin to assemble the entire speech back together again. So this shotgun sequencing technique has a number of really significant challenges. First of all, there's a pretty high error rate still in sequencing technology, and so we actually have to sequence the same exact pieces multiple times. What people usually aim for is about 10 times coverage. So each part of the genome is sequenced 10 times independently. Another problem is gaps. So you end up getting these contigs which have been assembled, but invariably they don't assemble into these long chromosomes. There are gaps, and those gaps occur at places where the DNA is very repetitive in nature. So you might find, for example, a transposon sequence at multiple locations, and it makes it hard to figure out which one will connect to which. There are some techniques to actually get around this problem of filling in the gaps, but this still remains a big problem. So in 2001, the human genome was sequenced, and there was a big splash in the news, and this is the cover of the magazine Science where it was reported. However, completion of the genome is really actually very arbitrary. So at that point in 2001, there were still 144,000 gaps in that DNA sequence. Even 9 years later, in 2010, there were still a number of gaps remaining, and to my knowledge, some gaps still remain that are just unsequenceable parts of the genome because they are so repetitive in nature. The sequencing of the human genome and many other genomes has now ushered in a new area of science that is called comparative genomics. And comparative genomics is a new research field that compares the genomes of different species. So people look at the sequences themselves, they look at the content, the function of the genes, and the organization of genes, and try to learn something about their research question. The monoorganism Drosophila melanogaster, which we've worked on in lab, was sequenced back in the late 1990s, and since then, a number of other species of Drosophila have also been sequenced. And people are very interested in using these sequences to investigate comparative genomics research questions. At this point, Drosophila melanogaster is really the only species that has been completely annotated. So we know where all of the genes are, and we know where individual exons start and end. The other species still need a lot of work, and that is what our project is going to focus on. The species that are indicated in green are species that our project has either looked at in the past or are currently looking at. So Drosophila melanogaster has a haploid number of 4. So each individual would have 8 chromosomes. The first chromosome is also the sex chromosome, and then there are 3 autosomes, what are called chromosome 2, chromosome 3, and chromosome 4. The research question that our project is investigating has to do with the 4th chromosome, which is also called the dot chromosome because it is very, very tiny. This dot chromosome has some really unusual properties. This chromosome is very small. It's also very transcriptionally silenced, and a couple of genes are able to escape that transcriptional silencing and be expressed at high levels. We're very interested in understanding how those genes can actually escape that repressive chromatin environment. So what our project is going to be is to look at this raw DNA sequence coming from a lot of these other Drosophila species and locating the genes on those chromosomes. We know that cells identify the location of genes in their genome based on their promoters. Humans actually have a little bit more trouble identifying genes based solely on the promoters. And next, we're going to go through and talk about what that evidence is that we can use to locate genes in this raw DNA sequence. So the project that we are a part of is something called the Genomics Education Partnership. This is centered out of Washington University in St. Louis. And it is a collaboration of a bunch of faculty at different undergraduate institutions that are getting undergrads involved in genomics research. So this core facility will take these pieces of the DNA sequence and break it up into what are called contigs. These are around 40 to 60,000 base pair chunks of genomic DNA. These are what we call raw DNA sequence, so we don't know what genes are on there at this point. And each school is going to claim a number of different contigs to annotate. Some of these contigs may be on that fourth dot chromosome. These would be the experimental projects. There are also projects from the third chromosome, which would be part of a control. So what we're going to be looking for is simply we'd like to identify all of the genes that are found in that raw DNA sequence. We are only going to be looking at protein coding genes. And in fact, we're going to ignore the rest of the parts of the gene that are the non-protein coding regions. So we are going to be ignoring the 5' and the 3' untranslated regions. We're going to ignore the promoters, enhancers, any genes that don't actually code for proteins like functional RNA genes. What we're going to do is we're going to look at that sequence and we'll be looking at the coding strand sequence for each gene. So the coding strand sequence will tell us what the messenger RNA sequence is actually going to encode in the protein form. And the results that we will look for are actually pretty simple. We're going to look for the very beginning of that protein coding sequence, the start codon, and we will find the exact coordinates in that 50 or 60,000 bases that correspond to each exon. Then at the end of the gene, we'll look for a stop codon. So we're locating the positions of all of the protein coding exons. Some genes may actually go down in number. So here we start at 5782 and the gene actually ends at around 4400. This gene will be on the minus strand, so it will be going in the opposite direction from a higher number to a lower number. We're going to locate genes based on evidence, and the evidence that we will use takes four forms. So first of all, we're going to look at following basic biology rules in order to locate genes. Second, we're going to look at what has been transcribed into messenger RNA. Third, we'll look at similarity of sequence with related species, and then we'll also look at computational evidence. This information is all going to be in the introduction to the gene annotation project in your lab manual. First, I'd like to talk a bit about the basic biology rules that we need to follow when we are doing gene annotation. So some of these are pretty straightforward. Genes are only on one strand of the DNA. They're not going to switch between the two strands. Exons are going to appear in order along the DNA strands. Coding regions have to start with a methionine. They have to end with a stop codon. And there has to be an open reading frame. This term, open reading frame, means that you have a stretch that has no stop codons in the middle of it. We'll also look for the consensus sequences for splicing. We've previously talked about the 5' splice donor as being a GT in the DNA or GU in the RNA, and the splice acceptor site as being an AG. We are not going to look for that branch point A, even though that's an important sequence in splicing. It is a variable distance from the end of the exon, so it's very difficult to actually locate that site. If you'll recall, in addition to these two bases on either end of the intron, there are also some other preferred sequences in the splice donor and the splice acceptor. Those are going to be taken into account by computer predictions. There are a couple of very important concepts that I need to go over here. So the first is reading frame. So we are given a raw piece of DNA sequence, and it's important to understand that that DNA sequence has 6 possible ways that it can be translated. There are 3 ways using the top strand as the coding strand, and those are shown here. So if you started from the very beginning and that CCC was the first codon, then the next one would be AAA followed by TTT. If you moved over 1, that would change the reading frame, and your first codon would be CCA followed by AAT. If you moved over 2 and started, that would give you yet different codons. This will be called the +1 reading frame. The second one would be the +2 reading frame, and then the third one is the +3 reading frame. We can also translate that DNA using the bottom strand as the coding strand. These are going to be called the minus frames, and the first one would be TTA. Frame -2, you would skip that first base and have the first codon be TAT. In the -3 reading frame, you would skip the first 2 bases and the first codon would be ATT. You will all be learning to use a software called the UCSC Genome Browser, and I'd just like to start to introduce that. So at the top of the window here, you'll see one strand of DNA. And this one is in fact translated into 3 different frames that are shown below it. The methionines are in green, the stop codons are in red. As you all know, DNA is double-stranded, but only the coding strand is shown in this view. If you actually click this arrow right here, it's going to show the reverse strand going 5' to 3' in the other direction. Another concept that I need to introduce is the idea of phase. So a particular gene is going to be encoded only on one strand of the DNA. So for a particular gene, it will either be in the plus frame or in the minus frame. Now, each individual exon can actually be in a different reading frame, and that is because when we remove the introns, these ones don't have to be in groups of 3. So you might have that first exon, for example, in frame +2, and the second exon might be in frame +1, for example. When the exons are joined together by the splicing out of the introns, they need to form an open reading frame. So they need to come together so that the 2 exons are both in the correct reading frame. When we do gene annotation, we're going to pay careful attention to how many extra bases are found at the end of an exon that are not part of a codon. So what you see here is an intron, and that is going to be cut out. You see the GT at the beginning and the AG at the end of the intron. If you look at the codons here, this first exon has these individual codons, and then there is one extra base at the end of the exon that is not part of a codon. We're going to call that phase 1. So the phase is simply the number of extra bases at the end of an exon that are not part of a codon. At the beginning of the next exon, we know that these ones, the CCC is a codon and the GGG is a codon. And so that gives us 2 extra bases, these 2 A's, that are not part of a codon in this particular exon. So therefore, this exon will be phase 2. It has 2 extra bases that are not part of a codon. When you splice those exons together, those extra bases need to form a codon. And so here, in phase of 1 would be compatible with a phase of 2 and to make a codon when it gets spliced together. Importantly, phases can either be 0, 1, or 2. And 0 is simply means that there is no extra bases that are not part of a codon. And so if you have a phase of 0 at the end of one exon, you will need to also find a phase of 0 at the beginning of the next exon. Let's take a brief look at the genome browser and identify what phase this particular exon is in. Here, I'm telling you that the end of the exon is right here at this blue line, and that GT is the splice donor, which is the beginning of the intron. This exon is being translated in frame +2, which is the middle line here. And so these amino acids, S, P, N, E, are the amino acids that are being encoded by that exon. If you go to the very end of that exon, what you'll notice is that there is one extra base here that will end up in the exon that is not part of a codon. Therefore, the phase of this particular exon will be phase 1. Another way that we locate genes on the DNA sequence is based on what is transcribed into messenger RNA. The idea is that if you find messenger RNA of that particular sequence, we know that has been transcribed, and that is pretty good evidence that there is a gene in that region. So there have been a number of experiments that have been done where messenger RNA has been taken from a variety of different tissues and sequenced. This kind of data is called either RNA-seq data or EST data. This is actually shown on the genome browser as these peaks. These particular regions are regions where messenger RNA sequence has been found that is the same sequence as that particular sequence on that contig. If we look a little bit closer, what we'll find is that if we look at a particular exon, we'll find that you tend to have very low levels of RNA-seq data, and all of a sudden it will jump up right at the beginning of the exon, and then it will quickly fall off at the end of the exon. This is because the intronic RNA is very unstable, so even though it is originally transcribed, it does not hang around in the cell for a long period of time. Another piece of evidence that we will use in gene annotation is looking at similarity with related species. And in fact, this is probably the most important evidence for the experiment we will be doing in lab. The idea here is that protein coding sequences are really the most important sequences in our genomes, and therefore, if you change them, they're very likely to cause deleterious effects to the organism. Thus, they tend to change much more slowly over evolutionary time than other sequences. And if we compare between related species, what we'll find is that the protein coding sequences are much more similar than the other sequences in that organism. We're going to use Drosophila melanogaster as our reference genome because it is very well annotated, and the sequences we will be investigating are fairly close evolutionarily to Melanogaster. There are several different types of conservation that are found. So first of all, we expect that the primary amino acid sequence will be similar between our reference genome, Drosophila melanogaster, and the related species that we are investigating. Second, we expect there to be a conservation of gene structure. So we expect whenever there's an exon in Melanogaster, there will also be an exon in our related species. So this other species will likely have the very same number of exons for a particular gene as our reference genome. Third, we expect that the exon length will be conserved as well. This has been found over and over that exon length tends to remain relatively close even if the amino acid sequence begins to diverge between different species. In order to compare sequences, we're going to learn to use a very important program called BLAST. BLAST stands for Basic Local Alignment Search Tool. There are 5 commonly used BLAST protocols. First of all, you might want to compare a nucleotide sequence to another nucleotide sequence. That would be BLAST-N. You can also compare a protein sequence to another protein sequence, BLAST-P. And then there are 3 other protocols which take nucleotide sequences and translate them in all 6 reading frames in order to compare the predicted proteins that could arise from a nucleotide sequence to either another protein sequence or another translated nucleotide sequence. In our project, we will use BLAST directly, and also the raw sequences that we will be annotating have already been BLAST searched against all the Drosophila melanogaster proteins. And here you can see an example of that. Wherever you have a little rectangle, you have similarity between the raw DNA sequence and a Drosophila melanogaster protein sequence. The final type of evidence we will use in gene annotation is computational evidence. So many people have attempted to write programs that predict the locations of genes from raw DNA sequence. These are called ab initio gene prediction programs. All of these programs assume that there are recognizable sequences that a cell can use and that a program could detect. There are multiple different ab initio prediction programs that we will use. Most of them have a very, very high error rate. Just as an example, I want to show you the results from one small section of a piece of DNA. So here on the side, you see that there are 5 different ab initio program results shown. 2 of these programs think that there are genes in this region. One of them thinks that there's one long gene with lots of different exons. The other one thinks there are 2 different genes. The other 3 programs actually did not find any genes in this region. So in summary, there are 4 types of evidence we're going to use. In evidence-based gene annotation, we're going to follow basic biology rules. We're going to use RNA-seq data. We're going to look at the similarity of the sequence with our reference species, Drosophila melanogaster, and we will use computational evidence.